Hello, everyone. In this chapter, we are going to address cancer. As we address this chapter, I want you to focus on a number of questions. The first question I would like you to ask is, what is cancer? The second question is, what are the characteristics of cancer cells? How are they different from normal cells in the body? What are the stages in the development of cancer? Another question would be, what is angiogenesis and what is metastasis? What are proto-oncogenes and tumor suppressor genes? What is their involvement in cancer development? Also, we want to ask, what are the different kind of cancers we know of based on the type of tissues where they develop? What are some reasons for the development of cancer? Both intrinsic reasons, meaning what is happening inside of the body, independently of what is happening outside. But then, what are some environmental conditions that can actually account for the development of cancer? Now, with all these questions in mind, we also want to ask, is there a way to prevent cancers? Is there a way to treat cancers? If so, what are these different ways? So these are few questions I want you to keep in mind as we start diving into this topic. So now, the first step is defining what cancer is. Cancer, it's basically uncontrolled cell division. Now, we talked about cell division or mitosis that occurs in somatic cells. We'd say that mitosis is basically in the body to repair some of the tissues that need repair, to replace all tissues, all cells, to perform growth in the body, to do development, to name only those different aspects. We also saw that during the cell cycle, the cell has to control how often it is dividing. Why? Because some cells can undergo some mistakes during the copying of the genetic information, a process that we call replication, right? Some mistakes can occur, some mistakes can be tolerated, other mistakes are otherwise just too detrimental to keep the cell alive. Some other mistakes are actually going to cause the cell to never die and multiply forever. That is the case of cancer. Cancer, it's uncontrolled cell division. Now, what is characteristics of these cells that divide without control? Most of the time, what we notice is that these cells typically are going to lose the ability to differentiate and to contribute in the function of that cell in the body. If you consider, for instance, a bone cell is supposed to be in the body to produce bone and to maintain bone, right? When a bone cell becomes cancer cell, it undergoes uncontrolled cell division. At that point, that bone cell that is now a cancer cell is not going to serve its original purpose anymore. It's going to lose the ability to fulfill that function. Now, other thing that we observe in cancer cells is that their nucleus in a cancer cell are abnormally large. Now, the fact that this nuclei are abnormally large is a sign that this cell is more active than it should be. which causes actually that cell to be metabolically so active and multiply that, that is supported by the enlargement of the nuclei. Now, cells are going to experience unlimited cell division, and that unlimited cell division, one of the ways that the cell undergo unlimited cell division, it's, for instance, when they express the gene that is responsible for the production of one enzyme that is called telomerase. Now, know that our chromosomes have at the very end, at the extremity of the chromosomes, a segment of DNA that is a repeated segment. That segment is called the telomere. Each time our cells divide, the telomeres shrink. The more the cell divide, the more the telomeres shrink. Now, there is a point in time when telomeres are so short now that the important genes in the chromosomes start being altered. At that point, that cell cannot multiply anymore. That cell will have to undergo apoptosis and die. And apoptosis being programmed cell death. We talked about this, right? Now, in a cell that is expressing telomerase, well, this telomerase is going to cause telomeres to not shrink. In fact, the telomeres are going to grow and be maintained, which causes the cells to become immortal, right? So here, at the very bottom here, we do have some cells that are actually going from being normal to becoming abnormal. Now, we can see in this cell here, we see how the nucleus is relatively small. The same tissue, after undergoing mutation, and the cells start losing their function. Now, notice how the shape of the cell is different in the mutated cell versus the normal cell. That's one. Two, the nucleus in the mutated cell is much larger. Now, if we come here in C, we see that the nucleus right here is occupying relatively most of the cells. In terms of shape, that cell has lost its normal shape, right? This is a typical cancer cell. Now, cells, as they undergo uncontrolled cell division, they end up forming a mass of cells. That mass of cells is called a tumor, right? Many tumors, it's, plural of tumor. Now, there are two kinds of tumors. You have benign tumors and malignant tumors. Benign tumors are these tumors that are encapsulated. They are closely attached to each other, forming a mass of cell. This tumor typically do not go in circulation. They do not go invading the rest of the body. However, malignant tumors are these tumors that usually are not encapsulated. In other words, the cells are not kept in one single mass. Cells have lost the ability to attach to each other and gain the ability to actually go in circulation, go in the bloodstream, go in the lymphatic tissues, and at that point, the cancer cells are going to spread throughout the body, which is actually a really bad news. Remember that these cells can divide without control, right? So wherever they go, they're going to start multiplying, crowd the space, use the nutrients, and this is going to cause a dysfunctioning in the body. So one other characteristic of cancer cells I want you to remember is that these cancer cells can actually divide without growth factors. So now, typically in the body, for our cells to divide, they receive some signals. And one of the signal, it's called growth factor. And that growth factor, it's what is going to trigger development, trigger differentiation, trigger multiplication in a healthy manner, obviously, right? But interestingly, in cancer cells, the cells are behaving as if they are receiving a signal from a growth factor to divide all the time, while the growth factor is not in fact released, right? So they are just stimulated and multiply all the time, which is a problem for our body. So another thing you want to know about cancer cells is that cancer cells do not become cancer cells all of a sudden. It's a process. It's gradual. It's multiple stage. So I want you to remember that cells, they become cancer cell in a gradual manner, right? It's a multi-stage process. Now, as the mass of cells grows, this mass of cell needs food. That food, where is that food coming from? The blood is the one bringing food, but also taking away waste, right? Now, blood vessels are going to grow into the mass of cancer cells that is developing so that this mass of cancer cells can have access to nutrients. Whenever new blood vessels develop in an area, we call this angiogenesis. Angiogenesis. Now, with angiogenesis, the cells are not only going to have access to food, but also they're going to lose the ability to attach to each other so that they are going to go in the bloodstream and start going into the remainder of the body. That is a stage of cancer that we call metastasis, right? This is not a good news. All right. So, what are the different stages in the development of cancer? I want you to remember that there are three stages. The first stage, it's initiation. The second stage, it's promotion. The last stage is progression. Three stages. In initiation, you have a normal cell that undergoes a mutation. That mutation is going to cause that cell to misbehave. But at that point, that cell is not a cancer cell yet, right? But it already has the potential to become a cancer cell. It starts undergoing somewhat uncontrolled cell division, but not completely wild uncontrolled cell division. Then we have promotion. Promotion, it's when the tumor develops out of that primary cell that was mutated. In general, a second mutation triggers that tumor development. So the cells are going to grow in mass. They are going to be mutated so that they are going to multiply more. Finally, we have the progression stage where the cell is going to be mutated again. So one mutation is not enough. Two mutations are not enough. In general, it's at least 3 mutations for a cell to go wild when it comes to uncontrolled cell division. For the cell to lose the ability to attach to each other, to lose their function, and to go in circulation and invade organs that are at a distant place compared to where the cancer was initiated. All right, so here we have a graphic representation where we show of all this epithelial cell that you see here, there is one in particular that has a modification, right? It has a mutation. Now, that cell is going to multiply, and unfortunately, out of this mass of cells, one of them can be again mutated. And this cell that is now mutated is going to even multiply more. And as it multiplies more, in the process of multiplication, because DNA has to be copied all the time, now the machinery that copies the genetic information can make mistakes, right? And that mistake can lead to a nasty cell, a nasty cancer cell. So that cell now is going to grow in such a huge mass that there is going to be a need for blood vessels to develop. These blood vessels and lymphatic vessels are going to develop to bring nutrients. And as these blood vessels and lymphatic vessels develop through angiogenesis, now these cells will start moving away from the mass because they have lost the ability to attach to each other. They move away from the mass, then they go in the bloodstream, in the lymphatic vessels, and at that point, they're going to be carried away from the site of origin and go cause a cluttering of some organs and organ systems far away from the origin of the cancer. All right, angiogenesis is essential to sustain the growth of cancer cells. Without angiogenesis, the growth of cancer cell cannot be sustained because they need food, right? But unfortunately, with that angiogenesis too, which is actually the formation of new blood vessels, well, these cells are going to gain the ability to go into the blood vessels and travel through the bloodstream. And metastasis, it's basically a stage during which the cells go in the bloodstream, go in the lymphatic vessels, and get distributed throughout the body. And again, that is a bad news. Now, what is the genetic basis of cancer? Now, let me tell you this up front. Any cancer, whatever cancer we are dealing with, has a genetic basis. I want you to remember that. And please write that down. Any cancer that develops has a genetic basis. Now, here is the genetic basis of cancer. During the cell cycle, there are a number of factors that are intrinsic to the cell, that are part of the genetic makeup of the cell, that control when the cell cycle should move on, or when the cell cycle should stop, or when the cell should die through apoptosis, right? There are two groups of genes I want you to remember. You have proto-oncogenes. Proto-oncogenes are these genes in your cells that, when expressed, they are going to stimulate the cell cycle to move on so that cells can multiply. All right? So while stimulating cell division, they are preventing cell death. They are interfering with apoptosis. So that's one set of genes. Another set of genes are the tumor suppressor genes. The tumor suppressor genes are very much what their name says, tumor suppressor. So they suppress the development of a tumor. In other words, they are going to stop cell division. That's their job, okay? Not only they stop cell division, but if some cells start multiplying more than what the body needs, these genes are going to be turned on so that these cells undergo programmed cell death or apoptosis. So I want you to remember that any cancer that develops is necessarily associated to either a proto-oncogene that got mutated and becomes what we call an oncogene, a cancer-causing gene, right? Or Another possibility for the cancer to develop is when a tumor suppressor gene is inactivated. Here is how I want you to think about proto-oncogenes. Proto-oncogenes are like, if you drive a car, you have the gas pedal. The gas pedal allows you to move the car forward, right? A proto-oncogene works like a gas pedal. The more you push on the gas pedal, the faster you go. If you never lift the foot off of your gas pedal, that's a problem, right? Chances of a crash is very high. By the same token, if you are driving and that your brakes, you hit them, but they are not braking, what is the chance that you can get in a crash, especially if you are on a highway, right? Think about that. The tumor suppressor genes, their job in the cell is to work as a brake. So when you push the brake pedal, your car has to stop. But if your brakes, there is something wrong with your brakes, you hit the brake pedal, it's not braking, where the likelihood that you're going to crash is very high, right? So think of the proto-oncogenes as the gas pedal, right? And the tumor suppressor genes as the brake in your car, right? If the tumor suppressor genes are mutated, the cell cycle cannot stop. Just like you cannot stop the car when your brakes are not working. By the same token, if you have a heavy foot and that your foot is on the gas pedal all the time, right? This is going to cause you to go faster and faster and faster and never stop, which can lead you to a crash, okay? All right. So the mutations that can occur in proto-oncogenes. And when these mutations occur in proto-oncogenes, the cell is going to behave as if it was receiving a stimulating effect of a growth factor all the time. Because normally, here is what happens in the cell. So the cell would receive a signaling molecule, right? That typically it's a growth factor that is going to attach to a receptor. By attaching to a receptor, that receptor is going to send a signal inside of the cell that says, well, we need to divide. So we have a signal transduction pathway that comes into play. That signal transduction pathway is going to ultimately cause the expression of some genes that are going to stimulate the cell cycle to move on. This is in a normal situation. Now, in the case of a proto-oncogene that is mutated, at that point, a proto-oncogene is not called anymore proto-oncogene, it is called an oncogene. Now these genes are going to behave as if the cell had received a signal that says the cell needs to divide, so that the cell is producing proteins that are essential for the cell division process to move on all the time, causing uncontrolled cell division. So that's one way of causing cancer, right? Now, also for the, here we have a situation where the tumor suppressor genes are inactivated. When the tumor suppressor genes are inactivated, these tumor suppressor genes cannot stop anymore, right? We have the production of a modified suppressor genes, or the product is not there at all, right? At that point, it's like you are not able to break, right? If you are not able to brake, your car is not going to stop. Okay, so this is what you need to see in this slide here, right? So one, proto-oncogenes are mutated so that they become oncogenes. Two, tumor suppressor genes are mutated so that they become inactive. All right. So when we consider cancer, we are going to name cancers based on the type of tissues where these cancers are developing. Oncology is the discipline of biology slash medicine that actually studies cancers. Okay? Oncology is the study of cancers. Some cancers occur in epithelial tissues. And where do we find epithelial tissues? On the skin, for instance, the mucous membrane, lining hollow organs, right? These cancers are called carcinomas. A carcinoma, it's a cancer that develops on the epithelium tissue. Then we have other cancers that we call adenocarcinoma. Adenocarcinoma are this carcinoma that develops out of glandular epithelial cells, right? For instance, when you consider breast cancer, you know that the mammary glands are some extension of the epithelium, and because they are an extension of the epithelium and they are glands, breast cancer, it's a type of adenocarcinoma, right? Then we have cancers that occur in connective tissues. Okay, when we talk about connective tissue, think about, for instance, the bones. Okay, bones is connective tissue. Cartilage is connective tissue. There are many more other types of connective tissues, right? This cancer of the connective tissues are called sarcoma. I want you to remember that, right? Sarcoma are also the cancer of the muscles. So not only connective tissues, but also cancer of the muscles. They are all called sarcoma. And then we have the cancers that develop in our fluids. What we mean by our fluid, our blood. Typically, if it's in the blood, it's going to be leukemia, right? That's a cancer of the blood. If it's in the lymphatic tissue, the cancer is going to call a lymphoma, right? That's a cancer of the lymphatic tissue. Please remember how we name cancers based on the type of tissue. I'm going to bug you with this, so please make sure that you remember this. All right. So the causes of genetic cancers, we already said that every single cancer out there is necessarily associated to either a tumor suppressor gene that is mutated or a proto-oncogene that is mutated, and now we call it oncogene, right? But the question we may ask is, do we know some examples of proto-oncogenes and some examples of tumor suppressor genes that are mutated so that cancer develops? That's a story we are going to cover in a minute. Before we dive into this, I want to tell you that beside the genetic causes of cancer, you can be predisposed to cancer and never develop cancer. Why? Because the environment plays also a very important role. Your lifestyle plays a very important role. Now, with that said, we can be in an environment that is actually going to trigger the development of cancer even when we were not predisposed to develop cancer. This is the case, for instance, when we get irradiated, right? Say I'm working in a nuclear plant, right? And I'm dealing with radiations all day long, all year long. that is actually putting me at risk of developing cancer. Other environmental reasons are carcinogens. Carcinogens are things like tobacco and pollutants, including heavy metals, right? That can be responsible for mutations in our DNA and therefore cause a risk of cancer development. Finally, but not least, and believe it or not, viruses are at the basis of many of the cancers that we struggle with, right? But regardless of the environmental reasons of cancer, it has to definitely boil down to an environmental condition that triggered a mutation in a tumor suppressor gene or a proto-oncogene, right? So that's basically how they interact. Now, what are the most prevalent cancers in male and female, and what is the mortality associated with these cancers? Now, between you and I'm not going to go through the list of all the cancers that you see here, but I want to call your attention upon a few things. In males, prostate cancers represent about 27%. Lung lung cancer represent about 14%, while colorectal cancer represents about 8%. Let's take a look at females. In females, breast cancer represent about 29%, lung cancer about 13%, and colon cancer about 8%, colorectal cancer. How about the number of people who are dying because of cancer based on the sex? By far, whether you are male or female, you have that lung cancer is the most prevalent cancer that is actually going to cause the death of people with cancer. And then you have in male, followed by prostate cancer. In breast cancer, in female, breast cancer is second. And then in male and female, we have the colon cancer, the colorectal cancer. Looks like this is now dead. All right. So now let's take a look at the genetic causes of cancer. Now, in the genetic causes of cancer, we are going to have, for the case of breast cancer, two genes that have been identified, BRCA1 and BRCA2. These two genes are tumor suppressor genes, right? This is an example of tumor suppressor genes that are responsible for cancer, right? The breast cancer. Another gene that is known, that has been identified, is the RB gene. That RB gene, it's a tumor suppressor gene that is associated with retinoblastoma. Retinoblastoma, it's a cancer of the eye. And then we have RET gene. The RET gene, it's a proto-oncogene that turns into oncogene after mutation, right? This gene, when it is mutated, it causes cancer of the thyroid, all right? So mutations in tumor suppressor genes and proto-oncogenes is going to predispose someone to cancer. However, that doesn't mean that the person is going to develop cancer. There are also some environmental conditions that play in the development of cancer. All right. Now, what are some other factors that play a role in cancers? We have transposons. Transposons are genetic elements in DNA that are able to jump from one place to another. These are genetic elements in our genome that are able to jump from one place to another. And as they jump from one place to another, they can actually insert within a proto-oncogene or within a tumor suppressor gene in such way that by inserting somewhere in a proto-oncogene, this is going to modify the gene in such way that that gene is going to be stimulated all the time. By the same token, it can insert in a tumor suppressor gene in such way that the tumor suppressor genes will not be expressed anymore. And if it's not expressed anymore, that's the end of that tumor suppressor genes doing its role, which is to stop cell division or trigger apoptosis. Now, the environmental condition, radiations, including UV light, right, in the sunlight, in tanning light, X-ray, can actually trigger a modification in the DNA that is going to affect a tumor suppressor gene or a proto-oncogene. You have some organic chemicals such as the smoke in tobacco. Well, the smoke, nicotine, right? That can lead to lung cancer, mouth cancer, larynx cancer, and many other cancer. We have some pollutants, including metals, right? Dust and chemicals, okay, that increase the risk of developing cancer. Viruses play a huge role in many of the cancers that we know of. Among the viruses, we're going to have viruses that is responsible for hepatitis B, so hepatitis B virus and hepatitis C virus. These viruses are going to lead to the development of liver cancer. The Epstein-Barr virus, this is going to cause a form of lymphoma, right, which is a cancer of the lymphatic tissue, right? The human papilloma virus that typically happens in female, right, occurs in female, can cause cervical cancer. The human T-cell lymphotropic virus, or HTLV1, this is going to be responsible for leukemia, and the HIV and the Kaposis sarcoma are going to be associated with the Kaposis sarcoma and certain lymphomas. Okay, HIV is going to cause some sarcomas, right, and can cause some cancers. All right. So when it comes to cancers, it is very important to screen the body, particularly when we know we are at risk of developing cancer. How do we know? We can know based on our family history, right? Now, in general, when it comes, for instance, to skin cancer, there are some changes that we see, but for other cancers, there are some also other changes that we see too. For instance, whenever we are developing cancer, typically there is a change in the bowel movement or the bladder movement, right? For instance, a prostate cancer, a male is going to feel the urge to urinate all the time, right? In a case of colorectal cancer, right, the bowel movement is going to change, right? A sore that does not heal, right? A sore that does not heal, usual bleeding or discharge. This is typical in cervical cancer in females. The thickening or lump in the breast or underneath of the testicles, right? We can feel that lump, right? Indigestion, indigestion, or difficulty in swallowing. That's typical of cancer of the throat. Obvious change in wart or mole, nagging cough, or hoarsening. So all the signs actually can be abbreviated caution, right? Caution, C for change, A for sore, U for unusual, T for thickening, I for indigestion, O for obvious, and N for nagging. But I'm not going to bug you with that, okay? I'm not going to bug you with that for the sake of the exam. I'm just sharing some trick with you here, right? So when it comes to skin cancer, typically we are going to have something on the skin that is asymmetrical, right? The borders are going to be irregular, okay? As you can see here, okay? The border are also irregular here, asymmetrical and irregular. We're going to see, in terms of color, we're going to see a variation, right? Between brown and dark black, right? That you can see here, okay? The diameter in general is going to be larger than about 6 millimeter, right? Which is larger than a dime, okay? And sometime we're going to see an elevation, E for elevation, and all these little things actually can cause us to think that, well, maybe there is the case of a skin cancer here, okay? Self-examination is very important, right? This is a picture, a micrograph of skin cancer. So we have in A, a normal breast, and in B, a breast with cancer, with a tumor, and you can see the tumor right here. So it is recommended that people do a self-examination on a regular basis by basically massaging a little bit the breast and the testicles, right underneath of the testicles, to see if there is an enlargement of something. For the colorectal cancer, a colonoscopy is common. This is not going to be a self-examination, obviously, right? You have to see a physician. But this is typically recommended when you turn 50 years old. Mammogram is also something that is recommended after 40. And the pap test, that is going to be for the cervical screening for the cervical cancer. Okay, so we have here the self-examination for a female, doing some rotation on the breast and massaging a little bit, just so you can feel if there is a lump or not, and if there is a lump, is that lump growing over time or not, right? You have the mirror check to kind of compare the two breasts and see if there is a difference in the size. Technically, there is always a small difference, right? But let me not go into the details of that. And for a male, just check right underneath of the testicles to see if there is a lump there. All right. So what are other ways to detect a cancer? There are many other ways to detect a cancer, and particularly by doing a molecular analysis of, of blood or tissues, right? So we have some tumor markers, and these tumor markers can be tracked when we do a tissue analysis or a molecular analysis, right? A blood test can allow us to identify antigens and antibodies, okay? For instance, the CEA, carcinoembryonic antigen, this gene is going to be very much expressed in a case of a colorectal cancer. Okay, so whenever CEA is in a large amount, in a in an individual who is at risk of colorectal cancer, there is a chance that that person is actually developing colorectal cancer. How about PSA? PSA, this is typically measured in males, right? Prostate specific antigen. It's going to be present in large amount in males that are developing prostate cancer. AFP, alpha fetoprotein, it's detected in the case of liver cancer. Other test, genetic test. So these genetic tests are going to test for the mutations in the proto-oncogenes and in the tumor suppressor genes. For instance, the RET genes, we can actually do a molecular amplification of the RET gene in an individual and look at the sequence of that gene and determine if that sequence is mutated or not. We have the P16 gene that can also be analyzed genetically, the BRCA1 that can also be analyzed genetically, and along with the genetic analysis, you are going to have a biopsy that is in general is going to be used to confirm the result obtained with genetic analysis. Now, what can we do to prevent cancers? Few things we can do to prevent cancer in terms of behavior, right? Lifestyle is going to be very important. The use of tobacco, you decide to smoke, you decide to not smoke, right? So this is a lifestyle behavior. Then we have sun bath and the use of tanning beds. You decide to use a tanning bed or not, but basically try avoiding that. Avoid radiations, right? Now, obviously, there are some jobs where you are more at risk to be exposed to radiation. So looking at the condition of employment is going to be very important. Be tested for cancer. Be aware of occupational hazards, right? That's what I was saying earlier. Be aware of postmenopausal hormone therapy, right? That can be very important. And also, vaccination can also play a very important role in preventing cancer. For instance, in the case of the HPV virus, where you can have a HPV vaccine. So, other ways to prevent cancer: have a right diet. So avoid eating too much to the point that you start becoming obese, right? Eat plenty of high fiber food. Increase the consumption of fruits and vegetables that are going to contain a lot of antioxidants and a lot of vitamin A and vitamin C, right? Reduce processed food, right? And that processed food is going to typically contain salt cure, chemicals, nitrite cured food, right? Like nitrite cured ham, salt cured ham. You have some smoked food. Now, obviously, I'm not saying that you should not eat any of this, right? I'm just saying that, the frequency at which you eat it, it's very important. Eat a lot of vegetables and really reduce the amount of alcohol consumption, because that can be a problem. In terms of treating cancer, now, you know that cancer is uncontrolled cell division. Uncontrolled cell division, it's a mass of cell. The best way to remove a mass is to do a surgery, right? Any other way is not going to be simple, regardless, right? So surgery is the removal of cancer cells. That is the best treatment that we can have when it comes to treatment of cancer. Any other treatment is going to have major side effects, right? Radiation therapy, which actually is going to be localized and target specifically cells. It's going to target the mass of cell that is undergoing uncontrolled cell division. But what you need to be aware of, it's that the very technology that allows to destroy the cancer cells is the same technology that can actually cause damages in normal cells, and unfortunately in some situation make the situation worse. But whenever we use radiation therapy, it's because we don't have a choice, right? Same thing for chemotherapy, right? Chemotherapy, it's a drug that goes in the system. As it goes in the system, the hope is that it's only going to target cancer cells. That's the hope. Nevertheless, cancer cells share more in common with your normal cells than they do share differences, right? I mean that they show differences, which means that, well, you are going to be at risk there of experiencing major side effects or side effects. Let's just put it that way. There are some cases of bone marrow transplant, right, to address some types of cancer. New therapies that are being tested or already active or actively used. You have immunotherapy. Immunotherapy is a kind of therapy in which we are going to modify immune cells and introduce these immune cells in a patient who is undergoing cancer. You have passive immunotherapy. In the case of passive immunotherapy, we have antibodies that are linked to a radioactive isotopes or a chemotherapeutic agent, and this chemical is injected to the patient. We have the p53 gene therapy. Now, p53, it's a tumor suppressor gene. That p53, that is a tumor suppressor gene, knows how to stop cell division and how to trigger apoptosis in the case of a need, right? So typically, in the case of that p53 gene therapy, the gene p53 is introduced in a retrovirus. By introducing that p53 gene in a retrovirus, the hope is that the retrovirus that goes in our system is going to go insert in our chromosomes so that an unmutated p53 is going to be expressed, right? So that now the mutated version is going to be compensated by the wild type version, the normal version. Then we have angiogenesis inhibition. This is basically preventing the development of blood vessel and lymphatic vessels. Okay, angiostatin and endostatins are two drugs that are commonly used to interfere with the synthesis of, the development of new blood vessels. So here we have an example of immunotherapy. Typically, a white blood cell that we call an antigen presenting cell is going to be obtained. And that white blood cell that is going to be obtained is going to be activated. We activate that white blood cell, and then, very importantly, we modify that white blood cell with adding a gene that is normal, right? So that gene that is normal is going to actually produce an antigen, and that antigen is very typical to cancer cells. So because this is an antigen presenting cell, what happens is that when that gene is expressed into a protein, which is now an antigen, that antigen is going to be presented right here on the surface. And now that the antigen is presented on the surface, this antigen is going to be recognized by our cytotoxic T cells. These cytotoxic T cells, in turn, are going to attack this these antigens. So any tumor cell that is presenting, that has an antigen on the surface, is going to be recognized by a cytotoxic T cell, and therefore the cytotoxic T cell is going to destroy all the cancer cells, right? This is a relatively complex technology, but it's a technology that is very, very useful. This is basically what we had for this chapter. Thank you for your attention. Bye now.